Good morning, everyone. I am Madhurima Datta, Zoology and Biology Educational Content Creators. Today's content for BSc, MSc, CUT, UG, and PG Csirnet Life Science. Paper Non-Coordinates or Diversity of Life Form, Unit Protista. And today I will discuss about the locomotion of the protozoa. Main locomotory organs are cilia and flagella. Amoebas are able to assume a variety of body forms due to flowing cell cytoplasm. The cytoplasm can be extended outward in pseudopodia of various shapes. Lobopodian are blunt-tipped, Filopodia are thin and sharply pointed. Rhizopodia are branched filament and Reticulopodia are branched filament that merge to form a net-like structure. And next is Exopodia are thin pointed pseudopodia that contain a central longitudinal axis filament of microtubules. Our short note is very important, that is amoebas that make shells are called testacid. Acrela and Difflugia have their delicate plasma membrane covered with a protective test or shell of secreted siliceous or chitinate material that may be reinforced with grains of sand. Next, I will discuss about the cilia and flagella. At first, structure. A cilium or flagellum has considerable internal structure. Each flagellum or cilium contains 9 pairs of longitudinal microtubules. It is 9 pairs of longitudinal microtubules arranged in a circle around a central pair. That is an axoneme. This 9 plus 2 tube of microtubules in a flagellum or cilium is its axoneme. An axoneme is covered by a membrane continuous with the cell membrane covering the rest of the organism. At about the point where an axoneme enters the cell proper, the central pair of microtubules ends at a small plate within the circle of 9 pairs. Next, kinetosomes. Also at about the point, another microtubules join each of the 9 pairs so that this form a short tube extending from the base of the flagellum into the cell. The tube consisting of 9 triplet of microtubules, which is known as the kinetosome or basal bodies. Next, centrioles. Kinetosomes are exactly the same in structure as centrioles that organize mitotic spindle during cell division. Centriole of some flagellates may give rise to kinetosome, or kinetosomes may function as centrioles. Next, mechanism of locomotion by cilia and flagella in protozoa. First, the current explanation of cilia and flagellar movement is the sliding microtubules hypothesis. So it is also known as the sliding microtubule hypothesis of locomotion in the cilia and flagella. The movement is powered by a release of chemical bond energy in ATP. Two little arms composed of the protein dynein and visible in electron micrographs on each of the pairs of peripheral tubules in the axoneme. And these bear the enzyme adenosine triphosphatase, which cleaves the ATP. Next, when bond energy is ATP is released, the arm walk along one of the filaments in the adjacent pair, causing it to slide relative to the other filament in the pair. Next, shear resistance causing the axoneme to bend when the filament slide past each other is provided by spokes from each doublet to the central pair of the fibers. The spokes are visible in the electron micrographs. Direct evidence for the sliding microtubules hypothesis was obtained by attaching teeny gold beads to axonemal microtubules and observing their movement microscopically. Next locomotory organs is pseudopodia. Pseudopodia are extension of the cell cytoplasm used in locomotion. This is the pseudopodia. The cytoplasm is not homogeneous. Sometimes peripheral and central areas of cytoplasm can be distinguished as ectoplasm and endoplasm. Endoplasm appears more granular and contains the nucleus and cytoplasmic organelles. Ectoplasm appears more transparent, that is hyaline, by light microscope, and it bears the basis of the cilia or flagella. Ectoplasm is often more rigid and is in the gel state of a colloid, whereas the more fluid endoplasm is in the sol state. Next, pseudopodia vary in composition and are of several types. The most similar are logopodian, which are rather large, blunt extension of the cell body containing both endoplasm and ectoplasm. Where a typical logopodium begins to form an extension of ectoplasm called a hyaline cap appears and endoplasm begins to flow toward and into the hyaline cap. Some amoebas characteristically do not extend individual pseudopodia but move the whole body with pseudopodial motion. This movement is known as the limax form. Next, phylopodia are thin extension, usually branching and containing only ectoplasm. They occur in some amoebas such as Euglypha. Next, reticulopodia are distinguished from phylopodia in the reticulopodia repeatedly rejoin to form a net-like mesh. Although some protozoologists consider the distinguish between the phylopodia and reticulopodia artificial. So it is a very important question, difference between reticulopodia and phylopodia. Next, members of superclass Actinopoda have axopodia, which are long, thin pseudopodia supported by axial rods of microtubules. The microtubules are arranged in a definite spiral or geometrical array depending on the species and constitute the axonium of the axopod. Next, axopodia can be extended or retracted apparently by addition or removal of microtubular material. Today's topic has ended.